Welcome to PSG College of Arts and Science. I am Dr. N. Geeta, Assistant Professor working in the Department of Zoology PG. And today's class we are going to study about the monoclonal antibodies. The monoclonal antibodies are very important and it has a lot of practical applications. And today's class we are going to study about how we can produce the monoclonal antibodies and its importance. And for diagnosing a cause or the root cause of disease is the major or key event which has to be prepared properly for the proper appropriate treatment. And for example, if you have a parasitic infection, the traditional method of diagnostic is to go for the direct microscopic examination or the culture medium or by detecting the antibodies in the patient's serum. But recent advanced technologies and as well as the basic understanding of antigen and antibody interaction, we have found out that even the antibodies can be treated to diagnose, to detect the diseases. And whenever we encounter an antigen, in response to that, antibodies will be produced by humoral immune response. And we all know that each antibodies which has a binding site for one of the epitopes of the antigen. And in this picture, you can see that a single antigen is having 3 types of epitopes, and the Y-shaped colored molecule is nothing but the antibody, and these antibodies are produced by 2 different B cells. So, single antigen, 2 antibodies are produced in our body, and that is called polyclonal antibody. The polyclonal antibodies, how are they produced means you can see in this image. For example, if an antigen is encountered, in response to that, different B cells will try to make a complex with the antigenic molecules, and the best or high affinity B cells which has the binding sites for that particular antigen will be selected by clonal selection. And what is the significance of monoclonal antibody is producing a monoclonal antibody is the key factor, because the polyclonal antibodies which are produced in vivo method can be used only for detecting the disease and it cannot be used for diagnostic purpose. Why it can't be used for diagnostic purpose means the polyclonal antibodies have low affinity towards a particular antigen and also due to heterogeneity. Heterogeneity means the same antibodies can able to bind with 2 different antigens. So, in that case, scientists have developed to culture the monoclonal antibodies. The monoclonal antibodies has to be produced by the hybrid cells in vitro method, which has to be cultured in the laboratory. And to have a monoclonal antibody, we need to have a hybrid cell. Why do we need a hybrid cell means the B cells which produce the antibodies has its own limitation. They have a very short lifespan. So, it has to be fused with the highly growing cell that is nothing but the cancerous myeloma cells. Why do we need to fuse the B cells with the myeloma cell means the B cell which is already differentiated and it became cancerous. So, the genes expression for the production of antibodies is already present in myeloma cells. So, only such cells can be fused to make a hybrid, and once hybrid is made, then it can be used under a culture medium and then it can produce the monoclonal antibodies. Let us see the outline of monoclonal antibody production. The first one is we need to immunize the mouse or any model organism to raise the antibodies. So, in such cases the mouse has to be immunized with 5 μg of antigen of our interest, and then we have to repeat the same process for many times for a good immune response. And after that, a week later, once the antibodies are produced in the mice, that has to be sacrificed and the spleen has to be aseptically removed. And after the removal of spleen cell, we have to do the enzymatic reactions to find out the viable B cells. Viable B cells are isolated from the spleen of the mouse, then it can be fused with myeloma cell. The myeloma cell as it is we cannot use, we have to select HGPRT defective or mutated myeloma cells. I will explain why we have to use the mutated myeloma cell in future. Okay. So, first myeloma, the cell has to be fused with the B cell and the resulting hybrid cell will be produced, and it has to be selected through the selective medium that is called HAT medium. The HAT medium is hypoxanthine aminopterin thymidine is the ingredient of the culture medium which has to be used to select the viable hybrid cells. And this picture shows the entire procedure of producing the hybridoma cell, and the technology is called hybridoma technology. And as I explained with this image, you can clearly see that how the mice are immunized and how the cells can be isolated by centrifugation method, and for each stage we have to do the screening process because we are doing something against the nature. So, we have to find out that whether the cells are under our control and it obeys the laws. And for screening process we have so many other tests, and by that we can find out the viable B cells or the hybrid cells in future which can be used to produce the antibodies. And this is the simple protocol. As I told that after immunization we have to isolate the B cells or the lymphocytes, and then it has to be fused with the myeloma cell which is already defective in HGPRT enzyme. HGPRT enzyme is very essential for the proper growth and the multiplication in the HAT medium because it is a defective one. Why we are using such a defective B cells and as well as the myeloma cell is we have to have only the hybrids and not the unfused cells. So, we need a hybrid cell. So, the culture medium will help to produce or help to isolate only the hybrid cells. Let us see what all the cells will be alive in HAT medium. Two cells will be alive in HAT medium. One is the hybrid cell, as we know that the culture medium supports the hybrid cells for its survival. And the next one is the B cell which already have a normal HGPRT enzyme, but due to short lifespan it can survive only for shorter duration and then it dies. And the cells which die in the HAT medium is the normal myeloma cell, that is simple cancerous myeloma cell. They cannot survive due to defects in enzymes. Next we go for the application of monoclonal antibodies. It has many applications and we can characterize to diagnostic application, therapeutic use, protein purification and miscellaneous applications. One is the diagnostic application, the very important, and day to day life we are using monoclonal antibodies without knowing that. You see, this is a home pregnancy tester and which has a monoclonal antibody. And here the monoclonal antibody is raised against HCG. HCG is a human chorionic gonadotrophin hormone which is produced by the embryo. So, when a woman is pregnant, only the fetus can produce the HCG. So, such HCG monoclonal antibody will be used to recognize or to test whether the person is pregnant or not. And in this image, you can see that HCG is bind with the monoclonal antibody and it makes the complex. And the next secondary antibody, which is also a monoclonal antibody, and that can recognize the complex of HCG and the primary antibody, which is already linked with the dye. So, that produces the color. So, that you can see in the window, you can see the two different lines. The first line is called reference line because we need to know that whether the tester is functioning properly or not, and for that you have a control antibody, and once the sample is exposed to the tester and immediately it shows the first line, that is in reference line. And next, if the person is pregnant, then HCG will be there and that can be form a second line, that is a blue line in this picture. So, by this we can sure about 99% the particular person is pregnant. And the next one is myocardial infarctions, the same, the death of cardiac cells. The heart cells, when they die, they release the myosin, it is a protein. When an antibody is produced against the myosin and that can bind the formation, so that complex, if it is already tagged with the enzymes, that causes the color changes by which you can easily diagnose. Second one is the therapeutic use. In therapeutic use, not only monoclonal antibodies are used for diagnosis, but also for the treatment. Hence, it is called a missile or magnetic bullets. Why it is called a magnetic bullets means, as like a bullets, it has a direct target and it can hit only the cancerous cells. And if the antibody is tagged with the direct drugs, then site directed drug delivery is possible, so that only the cancerous cell can die, the non-cancerous cells will survive happily. And monoclonal antibodies can be used for organ transplantation itself. For a patients, if they have any allergic symptoms to cyclosporine, that is the drug used for immunosuppression, and in that cases monoclonal antibodies which promotes the immunosuppression can be used as a good alternative which does not produce any allergy to the patients. So, the first antibody is MAOKT3, the first antibody recognized by USA FDA, and it is started administered to the patients as well. The next miscellaneous purpose is enzymes. Antibodies can be used as an enzyme, so that it can bind with the substrate and convert into a final product. And the other one is the antibodies called autoantibodies. Apart from cultural production, in our body naturally the antibodies unique for that particular person will be produced after the birth, and it reach its maximum level at the age of 2. So, such autoantibodies will be similar like our fingerprints, so which is so unique, so which can be used in forensic science. For example, if a suspect or the person who is involved in that crime area, if they get the samples, they can compare that antibodies from the crime area and with the suspect, and they can easily find out, and which gives us scientific proof that the person is whether involved or innocent to that crime. I hope this class given outline of monoclonal antibodies, and I hope this will help you for your further reference. And with this note, I thank you all.